Good evening, everyone, and welcome to the first week of PMRF NPTEL online course session. And my name is Nisha, and I'll be your PMRF TA for this course. So, the course is Drug Delivery Principles and Application. So, this is an archive course, as you all must be knowing, and all of you have joined this course. So, this course is being hosted by Professor Rachit Agarwal from IISc, and I work in his lab. I'm his student basically. I'm a PhD scholar here. So, what we're going to do in these sessions is these sessions are basically arranged for you to clear your doubts, as well as we'll be discussing some previous year uh, assignments, which would then give you an idea of what questions you're supposed to expect in the last certification course, the examination which will happen once you finish your course. So the way these sessions will be conducted is we'll first go through a summary of what all topics were covered in the first week as we go on. So this session will basically be held every week, and so at the start we'll just go over the whatever key points were discussed in first week, and then followed by we'll discuss the assignment. So does anybody have any doubt before we start? If not, then I'll go ahead. And let's try making the session a little bit interactive, wherein I'll just keep on asking you whether you remember this, whether you have learned this, whether you know this session. Hello, yeah, Vijay, are you trying to say something?
Yeah, ma'am. You please tell more about problematic sums now.
Yeah, sure. So most of these assignments will have numericals, at the end. So we'll be discussing those as well.
Yeah,
So
ma'am. Because of previous year, I will face lot of difficulties in final year exam, ma'am.
okay, sure. So we'll take care of that. We'll discuss as many numericals as we can from the previous assignment, as well as you people will also be getting new assignments in this course as well. So we can't discuss that before you submit the assignment, but we'll take it up in the next sessions. Is that okay?
But we want additional questions, ma'am.
Okay, sure. So I'll just browse the previous assignments, and we'll see if we can collect even more numericals for that. Is that okay?
Okay. Okay, ma'am. Thank you.
Yeah, yeah. Welcome. So I'll just share my screen. Can you people see the presentation in slideshow mode?
Yes, ma'am.
Okay, yeah, thank you. And just for your information, these sessions will be recorded because I'll have to upload it on YouTube. So this is also beneficial for you if in any case somebody misses one of the sessions and you want to go through these questions, you can just go into the YouTube link and you will get access to this. This PPT will also be uploaded and will be shared with you by the NPTEL team. So, and I apologize for this, but I'll have to keep on flipping in between the screens to admit more people. So just bear with me until then. Yeah, so we can start. So week 1 basically, so I'll just discuss some of the points which you might have already studied, just to give a recap of what was done. So first is the course starts with what exactly is drug delivery, its definition, and what is the purpose of it. So drug delivery is nothing but the appropriate administration of drugs for improving patient health. And the purpose of drug delivery or any drug delivery system is basically to give relief to the patient, either to completely cure the disease they are undergoing, or to lengthen the lifespan in case of a terminally ill person. Now you'll wonder of why. So most of the diseases that you see around have very well defined treatments. So then you'll wonder that why do we need even more drug delivery strategies in case we already have the treatments. So this is because you have to continually keep on improving the treatments, because there are several disadvantages of current treatments. For example, let's say some drug is given to a patient and then it has very severe side effects. So then it becomes difficult for the patient to continue with that treatment, because the side effects are so severe that you can't continue with the drug administration. Another disadvantage could be the longer duration of the treatment, or there could be frequent administration. So I can give you an example because I work with TB drugs. So in TB, that is tuberculosis. So in tuberculosis, the treatment duration is somewhere around 4 to 6 months, where people have to take drugs every day. And these drugs have very severe side effects, wherein you'll have vomiting, you'll have nausea, you'll have dizziness, you'll lose appetite. So then this becomes very difficult for the patient. So to reduce some of these disadvantages, that is why we are studying this drug delivery course, so that we can efficiently design these treatments. Now to do that, you need to know some of the basics of drug delivery, so that we can arrive at a proper solution for all of these problems. So just a minute. So as I said, some of the concepts that were discussed in week 1 are pharmacokinetics. So does anybody here remember what is pharmacokinetics? Can anybody define it to me? This is voluntarily, so I won't be taking any names for you to answer. If you guys want to just go ahead and give off an answer, it's okay. If not, we'll anyways be talking about it.
Pharmacokinetics is a drug distribute to all over the body, ma'am.
Right, yeah. So thanks for answering. What's your name? Because I can't see it on my screen.
Vijayaditya.
Okay, yeah, sure. Thanks, Vijay. So, yeah, that is one aspect of pharmacokinetics, as you said, drug distribution. So, pharmacokinetics essentially deals with, so once you administer the drug, the body is going to do a lot of things to that drug. So, whatever it does is what pharmacokinetics is. For example, it can be defined as, once you administer the drug, how is the drug going to be absorbed? How is the drug going to be distributed throughout the body? And then, since it's a foreign material for the body is going to try metabolizing this drug. So the drug metabolism will happen, and at the end the drug will also be eliminated from the body. So all of these factors constitute the field of pharmacokinetics. Now why this becomes important is because let's say you have a very efficient drug, you have a drug that is very very potent to cure the disease. Now you administer that drug to a patient and then you see that it is not having an effect. Now why it can't have an effect? Because let's say you have administered the drug and before even it can have its effect, the drug is very rapidly eliminated out of the body. Now despite having a very efficient drug, you are not able to help the patient. So all of these factors you need to know, how the drug is getting eliminated, what is the half life of the drug. So to do that, you need to study pharmacokinetics. Now there are various factors, parameters of the drug that can affect the pharmacokinetics, like solubility, hydrophobicity, or its ionization state. So some of the concepts further related to drug delivery are first pass metabolism, which is nothing but so when you administer the drug orally, this becomes very very, this is more concerned with when the drug administration route is the oral route. So what happens is when you administer a drug orally, most of the drug will be metabolized by liver even before it becomes available in the blood circulation. So that effect is known as first pass metabolism, because liver is very efficient in clearing of foreign material. Next you have something known as bioavailability. So what does this mean is, so you have given the drug to the patient. Now not all of that drug would be available at the site of action, because most of it, as I said, would be cleared off by the liver. So the amount of drug that becomes available for the actual effect at the site of action is known as bioavailability. This becomes important when you have to design the dose of the drug. Next you have elimination. As I said, the drug will also be eliminated, and further concepts related to elimination are, it can be a zero order elimination, wherein the how much drug is getting eliminated does not depend on how much drug is present in the body. So there is a constant rate of elimination. Then you have a first order rate. Then you have something known as half life. Half life is the time of the drug for it to be half of the drug that you have administered. In that time, half of the drug will be cleared. So that is known as half life. And there is something known as clearance, which is the clearance rate of at what rate is the drug being cleared out of the body. Next, so these are parameters related to pharmacokinetics. Next you have parameters related to the drug. This is also important when you are discovering new drugs, or you are getting new drugs into the market. So these are ED50, which is nothing but the effective dose 50. What this means is, let's say you have a drug and the ED50 value is the value of the drug or the concentration of the drug at which 50% of your population will see an effect of that drug. So all of these ED50, TD50 and LD50 are population parameters, because when you administer a drug, it is usually considered that you have a population of diseased patients and then you are administering the drug. So all of these will be considered in terms of population. So TD50 is nothing but the toxic dose 50, which means the concentration of the drug at which 50% of your population will see a toxic effect. Similarly, LD50 is the concentration of the drug at which 50% of your population will die, that is the lethal dose. And then there is something known as therapeutic index. So therapeutic index is nothing but the ratio of either TD50 or LD50 to ED50. So to have an efficient drug, what you need to have is the drug should have a very high therapeutic index, meaning it should be effective at a very low dose, but the toxicity level should be very very high. So that's how you see whether, once you have discovered a new drug, you think that it is potential, but then you have to characterize it using all these values. Then only you can go ahead with getting that drug into the market. Next, so all of this is the basics of pharmacokinetics. Then the professor goes on talking about prodrugs. So this is one of the methods of drug delivery. So basically prodrugs are nothing but these are compounds which are inactive, and when you administer it into the body, once they go into the body, they get metabolized and then they convert themselves into active drug. So now you'll wonder of why do we need prodrugs, because once you already have a drug, what is the need to go on making prodrugs out of it? So sometimes you need this for very specific applications, like let's say you have a drug and that is very hydrophobic, it is not at all dissolving in water. So now, and your body is made mostly of water, so you want the drug to be first soluble. If it's not soluble, it will not show its effect. So to make the drug soluble, you just convert it into a prodrug by conjugating it with some water soluble molecules or water soluble groups. Then it will become easier to solubilize the drug, and that will improve the bioavailability. Another application of prodrugs could be active targeting. For example, you want the drug to be active at only one particular site in your body. So what you can do is you convert it into a prodrug. Let's say you attach it with an ester chain, and this chain will only be broken down in presence of esterases, which are enzymes in the body which are present at specific locations. And once they act on this prodrug, they'll cleave this ester chain and they'll give off the active drug. So now this is where you can this is how you can design targeted therapy. Another concept is controlled release of the drug, wherein you want to achieve certain control over how much amount of, let's say you give the drug to the patient, and then you want to control how much drug would be available at the site of action, in what time, at what duration, or how long you should be doing this. So to achieve control release, you use something known as biomaterials. So biomaterials would be an extensive part of this course. Biomaterials are nothing but materials which you can use for drug delivery applications, which can be either synthetic or natural. We'll be talking more about biomaterials as the course goes on. And lastly, in the course, in the week 1 course, the professor has discussed about various polymers, which are class of biomaterials. So polymers are nothing but huge large molecules which are made of repetitive monomers, and polymers are very extensively used in drug delivery applications, as we'll see going ahead. And they are characterized, so there are various types of polymers which can be distinguished based on their chemical structure or their properties. Chemical structure meaning they could be homopolymers, which means that all of these monomers are the same in a polymer. They could be random copolymers. For example, there could be two different monomers making a polymer, and so forth. And based on properties, these could be thermoplastics or thermosets, which you must have seen in the course. And so to know more about polymers, we need to first characterize the polymer. We need to know what the polymer is, how it is going to behave once it's administered in the body, and so on. So to understand this, there are certain basic properties of polymers that we should all be knowing. First is the molecular weight. This is the most essential property used to characterize polymers. Then there is crystallinity, whether your polymer is crystalline in nature or amorphous in nature. Then there is melting temperature. Then there is something known as degradability. This becomes important because let's say you have designed a very efficient drug delivery system, and you have used these polymers and you have injected into the patient's body. Now you need to see whether you want these polymers to be present in the body throughout the patient's life, or you want them to degrade. So accordingly, whatever is the application, you need to tune in your polymers in such a way so that you can have an efficient treatment. So I've been talking for a really long time. Does anybody have any doubt here before we move on towards discussing some of the questions?
No doubt, ma'am.
Yeah, okay. Thank you. So let's go towards the questions one by one. I'll be going a little slow and give you time to read the questions on the screen. Can you see the question? So the first question is, which two statements are true for pharmacokinetics? So there are several options. So pharmacokinetics, PK studies help in determining dosage regimens. PK helps us understand the drug receptor interaction at the tissue level. It's used to identify new drug targets, or it shows how drugs are absorbed or metabolized throughout the body. PK studies how diseases spread in a geographical region, or it studies how drug dissolves in water and diffuses in air. So there are two right answers for this question. So take your time, read through the options, and let me know if anybody knows the answer.
Ma'am, can I tell the answer now?
Can you wait for a minute till the others can also respond, and then I'll give you. Yeah.
Okay.
I think most of you might have read through the question. So yeah, whoever was speaking, can you go ahead and tell me the answer?
For the answer, the first and fourth option. PK studies helps in determining dosage regime and as well as how drugs are absorbed or metabolized through the body.
Okay, that's one answer. Anybody else has a different answer or would like to volunteer with a different answer?
Ma'am, 1 and 3rd.
Okay, okay. Thanks for answering. So, yeah, both of you are correct. So, the correct answer is PK studies help in determining dosage regimens, and they also help us understand how drugs are absorbed. This is essentially absorbed. There's a printing mistake in the question. That's not absorbed. It should be absorbed or metabolized throughout the body. So, as we were discussing right now, pharmacokinetics essentially tells us how we should be designing our dosage regimen, wherein you decide where exactly you want the drug to be active, how much of the drug you want to be active, how often is the administration going to be, that is the frequency of the administration, whether it is daily, whether it is weekly or monthly, and how long is this going to continue for. So the rest of the options become irrelevant once you know what pharmacokinetics essentially does. So that's about the first question. Let me just check what is there. Yeah, shall we move on? I'll go to the next question then. So the next is, so you have been given a graph here, that is aqueous solubility of 3 drugs was tested at different pH, and we have to rank them in increasing orders of solubilities at pH 6. So take your time understanding the graph, going through the options, and then we'll talk about it.
Third option, ma'am.
Third option. Okay, okay. We'll wait for others as well. Thanks for answering.
First option.
Okay, somebody has first option as the answer. Okay. We'll see what is the right answer. If anybody else still has a different answer, so far we have got first and third. Yes?
Ma'am, second option, I think.
Second option. Okay.
Second answer.
Second. Okay. Anybody else? So far we have 1, 2, 3. Anybody has a different answer than this? Please make sure you read the questions correctly. It says increasing order of solubilities.
Ma'am, fifth option.
1, 2, 3, 4. Fifth. Okay. Tetracycline, chlorpromazine. Okay. 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 Let's see what the
It's third option.
third option is it?
Ma'am, eighth option.
Which option? Oh, the last one. Okay, the last one. Okay, okay. Yeah. We'll see the right answer. We are getting a lot of different answers. Okay, so the correct answer is tetracycline, indomethacin, and chlorpromazine, which is I guess the first option. The first option is correct. So let's look at this graph. So this graph is essentially aqueous solubility versus pH. So it essentially tells us what is the solubility going to be at different pHs for 3 different drugs. So the question is asking us that we have to rank them in increasing order of solubilities at pH 6. So you have to do, okay, just a minute. Yeah, all you have to do is you have to look at pH 6 and see which is the least soluble. So the least soluble is tetracycline, and then higher than that there is this graph cutting this point at pH 6, which is nothing but indomethacin, and then at the last you have chlorpromazine. So at pH 6 this is going to be your increasing order of solubilities. Is that making sense? So that's why the first option is correct. So people who have gotten second, third, fifth and eighth, is it clear now?
Yes, ma'am. We can proceed.
Yeah. Okay. Thank you. Can you, okay, somebody is asking, Wasim Akhtar is asking me to explain it again. So it's the same question, right? It was first, or which question are you asking me to explain? I'm assuming it is the second question. So I'll explain it once more. The others, please bear with me for a while. So this is nothing but an aqueous solubility versus pH graph. It tells us what solubilities these three drugs are going to have at different pH. So the question is rank them in increasing order of solubilities at pH 6. So what you have to do is first look at pH 6 of what is the solubilities of all these three drugs at pH 6, and then you see, since it is increasing order, you have to first select which is going to have the least solubility. So the least solubility here is tetracycline, because you see this curve has a dip at pH 6. Higher than that, then you have this curve which represents indomethacin, and then you have chlorpromazine. So that's why your increasing order of solubilities at pH 6 would be tetracycline, indomethacin, and chlorpromazine. That's why the first option is correct. Is it okay, Akhtar? I hope. I mean, you're not saying anything, so I don't know. I'm assuming it's clear. If it's not, we'll again come back to the question at the end so that others won't have to wait. Okay, thanks. Okay, so the next question is particles of what size can be excreted by kidneys? You have several different size range here, and if you guys have read through the lectures, you might be able to answer this. Hold on your answers for a while till everybody else can go through the question. Okay, does anybody want to volunteer for the answer?
10 to 100 nanometer, ma'am.
10 to 100 nanometers.
Less than 10.
Okay. One person is
Ma'am,
saying
option
third.
4.
I have one answer as the option third, and two people are saying option fourth. Okay. Anybody else?
Option 4.
Ma'am, it's
Okay.
less than 10 nanometers, ma'am.
Okay. Others?
Less than 10 nanometer.
Okay. Any other answer apart from option 4?
Ten to 100 nanometer, ma'am.
Okay. Another person is saying option third. Okay. Let's have a look at it. So yeah, the correct option is less than 10 nanometers. So we'll look at how kidneys actually filter any material that comes to it. So this is how the glomerular filtration membrane looks, which is present in the kidney cells. So you have this glycocalyx layer here. So you first of all have a blood vessel, then you have this glycocalyx layer, you have this membrane here, and you have these podocyte cells of glycocalyx. So if you've gone through the material which the professor has given, you'll see that the gap in this glycocalyx layer is around 70 to 90 nanometer, and then but if you see the podocyte layer, you have only a gap of 4 to 11 nanometer. So this is how the Bowman's capsule looks like. So the question is particles of what size can be excreted by kidney. So whatever is the size of your drug is going to be, it is finally been regulated by this 4 to 11 nanometer size. So anything less than nanometer will be excreted by the kidneys, but more than 10 nanometers will be, even if it passes from this glycocalyx layer, let's say you have a 70 nanometer particle, but it will be stopped by this podocyte layer. So the correct answer is less than 10 nanometers. Clear? I'll then move on. So the next question is, an antitubercular drug is administered via two paths, inhalation and IV. The alveolar fluid from lungs was collected at different time points. Please explain the large difference in the bioavailability of drugs for the different mode of administration. So there's a graph here which essentially tells rifabutin concentration at different time points in the BL fluid, and you have been given different options. So we have to guess at the correct answer, which will explain why the bioavailability is so different for these two routes. So take your time reading the answers, the options, and then we'll discuss.
Madam, what is the bulk fluid?
Sorry?
Bulk for bulk fluid.
Okay. So bulk fluid is nothing but the bronchoalveolar lavage fluid. So it's a method to collect specimen from your lungs. I'll explain what it is. So it is nothing but this fluid that is written here, the alveolar fluid.
Okay.
Yeah.
Okay.
Ma'am, sorry for the interrupt. You can
Sorry, I didn't quite catch you.
assignments, ma'am.
Sorry. Hello.
You can discuss current assignments, ma'am. You can discuss now in the current assignments, ma'am.
Oh no, so the current assignments I can't discuss until you submit the assignment, because then you're ideally expected to solve these assignments and you'll be graded for these. So that's why I can discuss it after you submit. So in the next week session I can discuss what the assignment that was given in the first week.
Yeah, okay. Thank you.
Yeah, welcome. Anybody knows the answer to this? You can start answering.
Ma'am, I think through the
The what? Not quite
Third
clear.
number.
Okay, the third.
Viability of inhalation.
Okay, one answer is third option. Anybody else?
ma'am, same, the third answer, inhalation route.
Okay.
Ma'am, third option.
Okay. Anybody else has a different answer? Okay. No? So we'll discuss the answer. Yes, all of you were correct. I think 3 people answered. So the correct answer is the bioavailability of inhalation route is higher for lungs and is hence reflected from the bowel fluid. So essentially what they've done here is they have given, so rifabutin is a anti-TB drug. They have administered the drug via 2 different routes. One is inhalation, one is intravenous injection, and then they get something, a graph like this. So essentially we have to look at the bioavailability. The question is, why is the bioavailability different for these two drugs? So as you must be knowing, bioavailability is nothing but the area under the curve. So you get it by plotting your rifabutin concentration at different time points, and then you have this graph, right? You have this curve. So whatever is under this curve, you basically take the area of this curve, and that is known as your and the ratio of that area under the curve for a different route versus the IV is known as your bioavailability. So if you do it here, you'll see that the bioavailability is very high. So essentially what this graph is trying to tell us, that if you give any drug via the inhalation route, that is going to have, so once you give the drug via the inhalation route, most of this drug is going to land up in the lungs, compared to what you give through the IV route. Because the IV route is given as it's IV, it will distribute all over the body, and very less amount of the drug will actually land up in lungs. So this question essentially tells us that, let's say if you're targeting lungs, if you want the drug, most of your drug to land up in the lungs, the best route to go ahead with is the inhalation route. Is this making sense? Is this clear?
Yes, ma'am, we can proceed.
Yeah, thank you. Okay, Then let's go ahead. The next question is, which of the following will have higher partition coefficient? We have certain compounds here and we have to identify which will have the highest partition coefficient. If you people know the concept of partition coefficient, it will be easy. Any answers? Any volunteers?
I think it will be option 1.
Option 1. Okay. Thanks for answering. Any other?
Option 1, madam. Option 1.
Okay. Thank you. You can call me Nisha. Please don't call me ma'am.
Option 4, ma'am.
Option 4. Okay. We have two different answers here, 1 and 4. Anybody else has another guess?
Option 1, ma'am.
Okay. Thank you. Okay, we'll look at the answer. So the correct answer is option 1. So we'll first look at what partition coefficient is. So it is a parameter used to characterize drugs. So partition coefficient is nothing but the amount of drug that is present in an oil phase versus amount of drug present in the water phase. So it's a ratio of these two values, log of these two values, and so basically the compound that is going to be more hydrophobic is going to have higher partition coefficient. For example, if you see all these compounds here, you see that in compound 1 there's a lot of CH2, CH2, CH2. So it essentially makes the drug very hydrophobic, and that is going to have more solubility in the oil phase. Let's say in that octanol phase. Oil phase is nothing but a polar any solvent. So compared to water, it will have more concentration in this phase because it is going to have higher affinity for this oil phase compared to water. So that's why this ratio will be very high for this particular compound compared to the others. So that is why the correct option is the first option. Is it clear? So once I'm in this slideshow mode, I can't see your messages in the Google Meet. So I'm sorry if I'm missing some of the messages. Somebody had answered for the kidney thing. So sorry if I missed it. Shall we go ahead with the next question?
Yes.
Yeah. Thank you. Okay, so the next is, which two statements are false for partition coefficient? So we just discussed what partition coefficient is. So see if you can go through the options and tell me the correct answer in a while. So please read the questions carefully. This is asking us which two statements are false for partition coefficient. Any volunteers for the answer?
I think it's the last option.
It's the last option. Okay, so you have to, so this is a multiple choice question. I mean, more than one option would be correct. So you have answered half, I guess. So 1 is the last option. Okay, any other answers?
3 and 5.
People are speaking at once. Maybe you can go one by one. Go ahead.
3 and 5.
3 and 4. Okay, 3 and
2
5.
and
2 and
4.
4. Okay. Anybody else?
2 and 5.
2 and 5.
2 and 4, ma'am.
2 and 4. Okay, we're getting a lot of permutations of this answer. I'll go ahead and discuss it. Anybody else has another answer before we go ahead? No? Okay. The correct answer to this question is the higher the lipid water partition coefficient, the lower the rate of transfer across the membrane, and higher the polarity of drug, higher the lipid water partition coefficient. So, I mean, just correlate to what you guys answered. So, we'll just discuss. So, as I said, partition coefficient is the ratio of how much the drug dissolves in an oil phase to the concentration of the drug in the water phase. So, the question is essentially telling us to identify what are the false statements about partition coefficient. So, if you see this, what is this? This is 3 and 5. So the correct option was 3 and 5. So let's look at the third option first. It says the higher the lipid water coefficient, the lower the rate of transfer across the membrane, which is essentially a false statement, because more the lipid water partition coefficient, more is your drug going to be hydrophobic. And if you know that the cell membrane is made up of lipids, it's already hydrophobic in nature, so it will be easier for your hydrophobic drug to transfer across the membrane. So if your partition coefficient of the drug is higher, it should the rate of transfer across the membrane should be higher. That is the correct statement about partition coefficient. Next option was the higher the polarity of the drug, higher the lipid water partition coefficient, which is also false, because more polar the drug, more if your drug is more polar, it is going to dissolve in the water more. So then this particular denominator will be more for a polar drug. More it will have more concentration dissolved in water, and as a result the partition coefficient will be a lower value for highly polar drugs. So is this clear? That's why these two statements are false. Rest of the options are correct about partition coefficient. Cool. If there are no doubts, then I'll go to the next question. So the next is, I believe this is the last question for today's discussion. Which of the following two statements are true for bioavailability? So we have several options listed here. We have to identify which two statements are correct for bioavailability. So read through the options and we'll discuss in a while.
Ma'am, does the question has multiple correct options
Yeah,
or
because
one?
yes, multiple correct options, because it says two statements, right? So you'll have to select two options.
Last option.
Okay, that is half of the answer, I guess. Anybody else?
Ma'am, second and last option.
Second and last. Okay.
Last
Thanks for answering.
second
Ma'am,
option.
fourth.
So okay, one person is saying fourth, one person is saying second and last. Anybody else?
Ma'am, fourth and last.
Fourth, 1, 2, 3, 4. Fourth and last. Okay. Anybody else?
Second and last option.
Second and last option. Okay, we're getting a lot of options. So yeah, let's look at the correct answer. The correct is as most of you answered correctly, it is the second and the last option. So let's look at the so as we were discussing, bioavailability is nothing but the ratio of AUC by a route of administration to AUC by IV. So first correct option is, it can be determined by calculating area under the curve by plotting serum concentration of drug over time. So to calculate bioavailability, what you have to do is, once you administer the drug, you collect serum and then see what is the concentration in the serum over time. Just like we looked for the last question, right? It was in BAL fluid. You can also do that for serum concentration of the drug. So that's one way, that's the way of calculating bioavailability. Next is bioavailability is considered 100% when drugs are administered intravenously. This is also a correct statement, because when you administer drugs through IV, it reaches each and every part of your body. So the bioavailability remains 100% when you are administering drugs intravenously. So the rest of the options are false. So people who answered maybe something else, you can get it checked. Is this clear?
Yes.
I think this is the last question I have for you. So there are 3 more questions in this assignment, and all of these 3 are numericals. But we checked and we found out that these 3 numericals are the same numericals which are in the current assignment. So I am not at liberty to discuss that until unless you submit, because then you'll already know the answers and everybody will get correct answers in the assignment. So I can discuss these 3 numericals in the next session. So that's all I have for you today. I have
Ma'am,
these
can you discuss similar questions?
similar questions, meaning?
Like similar to that assignment questions.
Okay. Similar to that, what exactly do you want me to discuss? Because I can't take up those questions until unless you submit the assignment. I will anyways be discussing it in the next. So I believe one question is polydispersity index, if I'm not wrong. So therein you just check what polydispersity index is, and first of all know what is the definition of that and how to solve that. You'll get it easily, those 3 numericals. I don't remember what the rest 2 are. So just go through those questions. I think it would be easy to answer. So once you submit in the next session, we can discuss that, and we can also discuss the new assignments once you have submitted those. So if anybody has missed some of these questions, I can if anybody wants me to repeat, I can go ahead. Does anybody have any other doubt? I've stopped presenting for a while. Any other concepts or any other topics which are not clear in the course, we can take those as well. If anybody has missed the starting questions when you have joined late, we can also discuss that. Okay, thank you, Dinesh.
Hello, ma'am.
Yes.
Can you explain about the volume of distribution? I have a doubt about in concepts.
Volume of distribution. Okay, I don't have a slide prepared for that. What exactly is your doubt?
The concept I actually didn't clear that much.
Okay, so volume of distribution, I believe, is nothing but the amount of drug that is present in the body to the concentration of that drug in the plasma. So volume of distribution will tell you where exactly. So based on that value, it's not an accurate value, it's an approximate value. So that value will tell us where your drug is going to be landed in the body. So that you can have an idea of once you administer where most of your drug will be concentrated. So if you have gone through the lectures, I think that there are a few examples there as well where one drug has a very high VD value. What that means is so the formula for VD is the amount of drug in the body to the concentration in plasma. So if this value is high, it means that most of your drug is present in the body and not in the plasma, in the fluid. It means most of this drug is present in the cells. Then you can have an estimate that maybe your drug is highly hydrophobic. That is why it is sequestering in all of these tissues and cells instead of coming in the plasma. If you have a very low VD value, it means that your drug likes to be in the plasma, in the fluid component of the body. So that way you can have an estimate of where most of your drug is going to land up. That is the concept of VD.
Understood, ma'am. Thank you.
Yeah, welcome. If anybody wants me to repeat the assignment questions, we can go through once again if people have missed the initial questions. Any other doubts?
I had a doubt regarding elimination.
Okay.
When we are if we have to calculate the total elimination of a drug, both the first order and zero order together, is that possible? Like how would you calculate the first order elimination while also taking the zero order elimination into account?
Okay, so these concepts, zero order and first order, will be specific to a drug. So your drug will either have a zero order elimination or it will have a first order elimination. I don't believe it will be together.
But zero order elimination is like the baseline amount of elimination, right? So that should be happening irrespective of which drug you're administering.
So some of these drugs are let's say you have alcohol. So when alcohol is getting eliminated from the body, it will not depend upon how much is present in the body itself. So it will have a constant rate of elimination. So irrespective of how much you have, it will be eliminated at the same rate. So that is essentially zero order. So I don't think any drug will have both zero order and first order.
Alright,
I'll just
so
yeah.
okay, so zero order elimination is not like the baseline level of elimination for everything.
No,
So
no,
it's
no.
just for
It's the
it's
rate at which it is getting eliminated from the body. So it's not
okay.
like a basal level of rate.
Okay, so it's very drug specific.
Yeah, it will be drug specific. But still, I'll go back and check if a drug can have both of these together, and we can clear it up in the next session.
Alright, thanks a lot.
Yeah, welcome. Any other doubts? If not, you can log off the meeting. If people have doubts, they can stay back and we can talk about it more. Otherwise, the session has ended. Thank you.
Thank you, ma'am.
Thank you.
Thank you, ma'am.
Thanks. Thank you.
Thanks for the class.
Yeah, sure. We'll meet next week. Okay, if that's all, I'll end this meeting. Thanks for joining. We'll meet next week as well. Thank you.